everybody. It's an honor, and I'm super excited to be here today. And I will tell you about some recent research that we've been doing in my lab. So I'm at the University of Michigan in the bioengineering department. So as Dr. Woodruff mentioned, I wouldn't be here without my absolutely amazing, inspiring mentors. Dr. Lonnie Shea, who is a bioengineer, and Dr. Teresa Woodruff, who is an endocrinologist and ovarian biologist, who taught me everything about ovaries and follicles that I know. So thank you. Usually when I start talking about my research, I always start with this cartoon from a paper published by Dr. Jackie Jervis, who was also a PhD student in Dr. Teresa Woodruff lab, and this was published in 2009 in New England Journal of Medicine that kind of summarized what are the available fertility preservation treatment and what are the experimental. So what you have over there in the rectangles is so potentially you could remove ovaries and then cryopreserve them, and then in the future you can either transplant them back or mature these follicles outside of the body in vitro and get to the mature egg. So my lab is working in these two directions, trying to actually make it clinical and not just experimental. So when I joined Dr. Woodruff Shea collaboration, this team already figured out that culturing follicles, which are three-dimensional structures on a 2D flat surfaces, is not really going to work. And even though it's working for mice and lower mammals, it doesn't really translate to non-human primates or humans or even cows and sheep. And the major reason is follicle is a three-dimensional structural and functional unit of the ovary. So you have to keep all the cells together. The oocyte has to be connected to the granulosa cells. And when you put them on the 2D surface, as shown here at the bottom, the granulosa cells migrate away and kind of leave the oocyte for its being. So what was discovered in that collaboration is if you take the follicle, you encapsulate it in a three-dimensional hydrogel, you can really support this three-dimensional structure. So the gray circle around the follicle is an example of a hydrogel. So when I joined their collaboration, they already were working with alginate, which is a physiological hydrogel, and it supports follicle development. And there was a lot of work done in that area. So we figured out how to culture secondary mouse follicles, got live births, and then through my work, we even translated a little bit with the non-human primates and worked with Dr. Mary Zielinski to promote non-human primate follicle culture from the primary stages all the way to the blastocyst. But then once we've done some important work with the secondary follicles, it was still difficult to culture smaller follicles like primary follicles and primordial follicles. So starting in my lab, I kind of focused more on that one. And then one of the observations that was done by Jessica Hornick, who was a postdoc in Teresa Woodruff lab, was that if you isolate primary follicles, and like you can see over there, day 0, so primary follicles in diameter less than 100 micron, they don't really survive nicely, either in 2D or 3D. They just die. And as you can see on day 8, you kind of see very shrunken, sad oocyte, and the follicle doesn't survive. If you co-encapsulate these small follicles in groups of 5, they actually survive better. Not necessarily 100%, but better. They don't grow as much, but they survive. But then if you put them in groups of 10, they just thrive, right? So they grow, they develop, they can be matured. And it was really exciting to see how, so green is 10x and 5 is 5x. So you can see that 5x groups survive about 50%, 10x up to 80%, and then the 10x cultured follicles reach larger size and can be matured. So our question was, as observational, it's so exciting, and because they're not touching, there's probably something that's exchanged between these two follicles, and it only happens when you co-culture them in groups of 10. So we asked this question, so what is it that they're exchanging? Because, I mean, it would be interesting. So we measured some of the cytokines in the media. So here you have the heat map. And then in this heat map on the left, so this bar, all these bars, so whatever is green on the bottom is the 10x follicles, and then the right is the 5x follicles. And you can see it's completely different signature of secreted factors. And then if you group them using principal component analysis, you see that 5x follicles group completely different than 10x. So the question was, is it additive or synergistic? So additive effect would be 1 + 1 = 2. Synergistic effect would be 1 + 1 = 5. right? So to answer this question, we said, well, if you just double the concentration of what 5x follicles secrete, you don't really get what the 10x do, and they kind of group completely separate here. And then the last column here also shows you that it's a different signature. So obviously, there is a synergistic effect. So once you reach certain concentration in those 10x follicles, something happens, and these follicles can actually survive and thrive. So we discovered and we concluded that there is a synergistic secretome effect. But then we also wanted to look at the transcriptional behavior of these follicles. So using TRACER, which is transcriptional activity cell array developed in Dr. Lonnie Shea's lab, we designed DNA segments. So basically you take a small segment and it's transcription factor binding segment, and then you connect it to the promoter, and then you connect this to a gene, this is fluorescence gene or luciferase gene. So what happens, you load this construct into a virus, and then you expose follicles to these viruses, and then when the virus infects the follicle, the granulosa cells mostly, with this segment, what happens, it kind of translates and it enters the genome of the granulosa cells. And then if you have transcription, specific transcription factor, and you can choose whatever transcription factor you're interested in studying. So when this transcription factor is present in the cell, then it binds to the segment, and then the promoter gets activated, and you have the production of luciferase. So if you add luciferin and the transcription factor is present, you will see glow. So we designed a few of these segments. So we wanted to look at GLI, HIF1, NF-kappa B, and some other transcription factors that we knew would be important for folliculogenesis. So we designed these genes, and then we infected follicles. So no one infected follicles before us. We were worried, but it worked. So we infected primary follicles, and then we co-encapsulate them in groups of 5 and 10. We culture them, and the benefit of this technology is that you don't have to sacrifice the culture. So you can just do it for the longevity of the whole culture and culture them for 12 days without missing and kind of following each follicle individually. So we could see the light, and we could, so this is the luciferase and the active, and you can see where it's actually expressed. And then you can also quantify. So the quantitative analysis was important for us because you can really quantify which genes go up and down over the time. So, and it was very interesting to see that, so again, green is 10x and red is 5x. That the 5x follicles are kind of delayed. So here you have the transcriptional activity of HIF over the time of the culture, and then you see HIF peaks 2 or 3 days after it peaks in 10x. And then the same thing is happening with NF-kappa B. So the 5x peaks somewhere 2 to 3 days after the 10x. So there is definitely another support for our idea that there is some sort of synergistic sequence that's happening. So from here, we're continuing identifying what are the factors that are important. So from the analysis of the cytokines, we are trying to figure out which have more weight versus less. So we identified 4 factors: VEGF, follistatin, MCP, and KC, that are mostly secreted and the most differential cytokines between the 5x and 10x. So we designed basically a rescue cocktail because the strengths of the system that we're using to measure those cytokines, that you can measure the concentrations, not only see transcriptional activity, but you can actually learn how much is there. So based on what we measured in vitro, we supplemented our cultures with all these 4 factors. And my student, Andrea, she has a poster working on this. So we decided to compare follicles individually encapsulated, less than 100 micron follicles, cultured in either supplemented media or conditioned media from 10x follicles. And then so we cultured them, and we didn't solve the problem. So the control follicles obviously die. They cannot survive individually. The conditioned media follicles really do well. So they like the 10x media, so that doesn't necessarily has to be the crosstalk between them, so just enough to get those factors. But obviously we're missing some factors because just by adding these 4 factors wasn't enough. So some of them do survive, and they look better than just individually, but it's not enough. So we're back to our drawing board, and Andrea is doing a lot of work, doing microarrays and doing some important computational biology to identify what are the factors, because our goal is to create this rescue cocktail that we can take each individual follicles and take it all the way to the survival. So this kind of summarizes my in vitro work. I also want to talk to you about the transplantation work or the in vivo work that we're doing. So I don't need to do a lot of introduction, and I actually appreciate the comment that was done at the end of the previous session about the importance of hormonal function in young girls especially. And with the numbers of these girls who had cancer in treatment before puberty is going up, I think it's very important to understand what these girls are missing, not only in terms of fertility, basically being able to have biological children, but also in being able to undergo physiological puberty. So this photo was published in Nature Outlook in 2013, and I think this is when I started my position at the University of Michigan. When I saw it, I was like, oh my God. So these two women are identical twins. Right, so the woman on the right had leukemia when she was very young, and obviously you can see that they don't look identical anymore. And there is a lot of different things. So she's shorter, and she's predisposed to diabetes and cardiovascular disease, and her bone health is not great. And unfortunately for the girls who lose the ovarian function before puberty, the only option to induce puberty and make them grow and develop secondary sexual features is to give them hormonal replacement therapy that was designed for postmenopausal women, delivers only two hormones in a steady state without any crosstalk between the hormones or the gonadotropin circulating in the body. And I think we kind of underestimate, or maybe we used to, not anymore, which is very exciting, but I think we used to underestimate the importance of the ovary and really the crosstalk that ovary has with fat, right? So we know that girls don't start puberty if they don't have enough fat in their body, and it's because they sense that the ovaries are ready to start growing, and obviously bones crosstalk with the ovaries and brain. So brain development during puberty is enormous, and I think when we just deliver steady-state hormones, there is no crosstalk, right? So our idea was, what if we take donor tissue from deceased patients or patients who donated their ovaries, and we encapsulate this donor tissue in immunoisolating capsules. So I'm a hydrogel person. I can make hydrogel from anything and for anything. So we took, sorry, thank you, Teresa. So I wanted to walk around. I just need to talk louder. So we took a donor ovarian tissue, encapsulated in capsules. So capsules have pores, and they allow diffusion in and out without the need to revascularize it. So it's going to be subcutaneous implant because it's only for the hormones. We don't need to think about fertility. Subcutaneously in the arm, it is self-regulating. I'm not really worried about any adverse effects because the ovary is going to respond to what's there. It's not going to be any kind of overdose. It's going to come from donor ovarian tissue. So from one ovary, I can make hundreds of these implants because we just cut it up into small pieces. And it's an immuno-isolating capsule. So you can use donor ovary, but you don't need to use immunosuppressive therapies. So how do you engineer this kind of immuno-isolating capsule? So it has to allow expansion because we know follicles grow and they need some space to expand so they can make more cells to produce more hormones. And then it also needs to allow diffusion of nutrients, oxygen, and also all the gonadotropins and release the hormones out. And it has to prevent rejection in terms of prevent immune cells such as T cells and B cells to actually get through the capsule. So in our separate project, we developed a hydrogel. It's based on polyethylene glycol. And you can see here, so it's a transparent, degradable hydrogel. Cells secrete proteases, it breaks down. And we use this hydrogel to isolate and encapsulate individual primordial follicles, implant them into the mouse, and then kind of follow this development. And you can see on day 0, we have individual primordial follicles, and then we put it in the ovarian bursa of mice. And then you can see over time how these tiny dots, they develop into large, growing, beautiful follicles. These gels get revascularized, and we see ovarian function. So gels, if we don't implant anything, the FSH, the ovarectomized levels in mice of FSH kind of sustain and persist. And then if we implant follicle with the hydrogels, the FSH goes down, and we maintain this nice distribution of the follicles. So we have primordial follicles in the beginning, and there's, after 2 months in the mouse, you see how the number of primordial follicles decreases, but instead you get more activated follicles that grow and secrete hormones, which is wonderful. But this is a degradable gel. So if it's degradable, it means that it's going to fall apart eventually, and the blood vessels will infiltrate and reject it. So what we've done, we encapsulated the ovaries in this degradable gel. So these are just pieces of mouse ovary. They're surrounded by degradable gel. And then we have a layer of non-degradable gel. This is how it looks under the skin of the mouse. And then this, you can see that the ovarian tissue here pointed with the blue arrow is in the middle of this gel. And it keeps the follicles and the ovaries surrounded and isolated from the host. So the first thing to test was isogenic transplantation, right? Even without thinking about the rejection, we're putting it in a larger capsule, so can follicles sustain? So just using the same strain mice, we could show that the follicles develop and they grow and they're fully isolated in this big bulk gel, but they still get enough through diffusion to grow and secrete hormones. The next question was, can this capsule, while sustaining the follicle development, can it still sustain itself in the allogeneic model? So we used white mice, biopsy mice, and we used their ovaries and implanted it in black mice. So first we wanted to test whether ovaries, because these are tiny ovaries, do they even reject? Maybe they don't even cause any immune rejection. So if you take ovary from biopsy, and you implant it subcutaneously in a mouse, it rejects in 3 weeks. So you see there is an increase in circulating allo IgG. It's fully gone. And then if you do CD8 plus staining, which is cytokine T cells, it's completely gone. But then if you encapsulate these ovaries in immunoisolating device, there is no circulating allo antibodies. So there is nothing. You can see here. And if you section, there is no infiltration of the graft, and then the mice continue cycling for, this is 14 weeks, but now we have data for more than 6 months. And then the follicles are there, and they look nice, and they persist. So we were super happy with this result. But then the next question was, and you all know this, right? So we'll take a small piece of ovarian tissue, we will implant it. It has to be small because everything is sustained by diffusion. It's going to exhaust itself. So a year or 2 years, I don't know how long it's going to last in humans, we'll have to go back and implant another one. So the question we ask, what happens if I have repeated implantations? Because possibly I can expose the host to this immune antigens. And then if we implant the second one, the immune reaction is going to be stronger and it's going to kill the implant. So if we, again, so the control is ovary. Ovary, non-encapsulated, you see how it just flares up. After the first implantation, you have about 1,500 circulating antibodies. After the second implant, it even goes up, so there's consistent memory response. But if you do it in immunoisolating capsules, or the first implantation, you really don't see much. And this is the quantitative analysis after first implant. And then if you implant the second one, there is really nothing much happening. So it's either the capsule completely blocks anything out, or maybe the antigen that can actually seep through don't really induce any kind of allo response. And in C, this is the implant that this is the first implant retrieved after the second implant. So this one was inside for 120 days, and we still see some nice antral follicles. And this is the second one, and there is no really IHC staining that can support rejection. That was very exciting. And then the last question that we wanted to ask was, what if we stimulate or synthesize the host before, right? Because humans are not mice. We are synthesized already. So we wanted to synthesize the mice. We implanted the ovary first, really created a lot of circulating allo antibodies. And then after this, we implanted immunoisolated ovary. And so this is the first ovary implant, not encapsulated, followed by encapsulated. So you see there is no real increase in the ovaries have, so the implanted encapsulated ovaries have the follicles and they sustain function. So at this point, we've done everything possible we could do with mice. And the next question was, what do we do next, right? Because I do truly want to take it to the patients. So I was very fortunate. We applied for pilot funding to do a pilot study with non-human primates, and we were able to go to UC Davis, and we had 3 monkeys, and we implanted them with the device. And then the second part was a little bit with human tissue in mice, and I will show you both. So when you try to translate human mouse ovaries into non-human primate or humans, there's obviously size issue, right? So Jean demonstrated in the morning, and so non-human primates follicles grow so much larger. So if you have to encapsulate it in a hydrogel, you really have to make sure that there's enough space. So we made our device larger, and then we went to the UC Davis, and we had 2 baby monkeys. So they just started their cyclicity. So this is their progesterone and estrogen levels before they were overreactomized. So they had some progesterone and estrogen peaks. And then the veterinarians ovariectomized them, and then we implanted the encapsulated ovarian tissue allogeneic. So we switched, we made sure that these animals were not sisters or related, and then we switched this, so it was allogeneic transplantation, and then after some time we start seeing those peaks. And I showed these results to Mary, Dr. Zielinski, yesterday, and she said it's okay to show. So yeah, it's okay to show. So we do see some potential promising peaks here, and then at the section we see nice follicles. Definitely one pilot study is not enough, so we do need to do more animals, but we were super excited to see that the ovarian, the non-human primate ovarian tissue could function in live monkeys. And then in terms of immune reaction, there was no difference between self and allo reaction, so it was very exciting for us to hear. So for 6 months and then followed by 4 months, there was no response. And this is how the grafts look like. So the ovarian tissue is right in the middle, and then it's surrounded by the hydrogel that doesn't get infiltrated by blood vessels. So this is our pilot, and we're trying to plan for a bigger study that we can actually follow development and health of these animals. And on the parallel kind of project, we start getting some human tissues from diseased patients. So in the lab, we would get the whole reproductive tract, and it's kind of either shipped on ice, and it's downtime less than 10 hours we're trying. So we remove these ovaries, we section them, as I learned in Dr. Woodruff's lab, very thin, so 1 by 1 centimeter, 500 micron thick. We cut them into small pieces, and then this is how my lab looks. And the tissue, for some reason, always arrives around 1 a.m. or 2 a.m. in the morning, and then they just keep working. I'm just there like, yeah. And then so it looks a little bit chaotic, but yeah, they managed to take care of all these tissues. So we vitrify. We learned this procedure, how to vitrify ovarian tissue. We also do slow freezing. We also do fresh implants, transplants. And then we look at the ovarian recovery after these freezing procedures. And then we encapsulate this ovarian tissue. We either implant it as it is to kind of have a control and make sure that capsule doesn't affect any ovarian development in these implanted tissues, or we encapsulate it in those hydrogels, and we implant it in immune-compromised mice, right? Because at this point, I don't think we will ever use xenograft, so I don't think at this point we will ever implant cow ovaries in humans. However, never say never, so maybe. But right now, we don't want to deal with xenograft rejection, so we're using skid mice just to see if the follicles can survive in these hydrogels. And we just started, so Margaret and Artie, they're helping with this project, and Margaret and Artie have both posters here, so I hope you had a chance to look at this. So this is how the capsules look under the skin of the mouse, and retrieved after 6 months, I think. We still see some nice surviving primordial follicles, but obviously we just started translating it into human or non-human primates. But then really trying to push forward, we applied to NSF. We got NSF I-Core grant. It's a small grant that allows us to travel all over the country and really do customer discovery. So I talked to a lot of people here, so I don't know if you remember, recognize me, but we bugged you. So we asked physicians and clinicians and people who deal with these cases, what do you want to do? How can we improve treatment of these girls and inducing physiological puberty? So we interviewed these people, and then we also contracted an outside company that performed a preclinical and clinical gap analysis, product development plan, clinical trial protocol, and we are right now in the pre-IND stage to apply to FDA and actually get started on the clinical trial. So I would love to talk to you if you have any ideas how to help us. I do want to highlight one more amazing project in my lab, and this is in collaboration with Dr. Malovic. It's actually, I'm just kind of around her genius. But it's a very exciting project about looking at the effect of testosterone on transgender youth and the ovaries. And Hadrian and Gillian, they have a wonderful poster here looking at, so they're actually looking at the rete ovaria. I don't know if you know this. We just discovered it. It was so cool. But we see that rete ovaria also changes when testosterone is administered to female mice. We just published the paper in Human Reproduction describing this model. So please check this out. To summarize, we do in vitro work trying to understand the synergy of growing small follicles. We are very excited about our immunoisolating capsule. We're really looking forward to try to take it to the humans, and we're also looking into ways to implant and revascularize isolated primordial follicles. Didn't talk about this, but we're also trying to build an artificial ovary. This is my wonderful lab, my wonderful collaboration, and I'm grateful to the funding. And thank you for giving me the opportunity. Thank you.